Hi everybody. I miss you guys so much and I'm so sorry that we cannot meet in the classroom. But since we can't, I did find a way that I can talk to you with our slides through YouTube. So I hope you guys like hearing the lectures this way. And I do look forward to hearing from you guys by email, also by phone. I've given you my phone number, my email. Feel free to contact me any way you can and hang in there, do your best. We're going to work together. Um, our next chapter here that we're going to start on is called the cell cycle, and I only have 15 minutes for each video, so I'm going to have to go pretty quickly here. I'm just going to touch on some of the main points I really want you guys to focus on and that you need to really look over for the tests and the quizzes and the main points of the chapter. That being said, if you have any questions at all, don't hesitate to ask me. There may be some things that I don't get to spend a lot of time on. Okay, so the cell cycle. The cell cycle is going to talk about the orderly set of stages from division of a eukaryotic cell to the time resulting in two daughter cells. Now, what does a cell do during its life? What is the cell cycle? Well, there's two major stages of the cell cycle. The first one is interphase, and it's going to include several different stages that we're going to talk about. And then there's the mitotic stage, which is going to include mitosis and cytokinesis, and we'll talk about both of those as well. Hold on here. Okay, so this is the diagram that you guys are going to see when we talk about the cell cycle. All of this up here in purple, I hope you guys can see my cursor. All of this up here in purple, this is the interphase. And this is going to be a cell just being a cell and doing its job for a lot of the time. So like a liver cell being a liver cell, or a skin cell being a skin cell, or a blood cell being a blood cell. And then just prior to that cell dividing to make a new cell, it's going to grow larger, it's going to double the number of organelles, and the DNA is going to be replicated, all of that within interphase. And we're going to talk about each of these individual stages as well, and it's also these stop signs that you see here, they're very important. And then you will also see in blue down here is the mitotic stage of the cell cycle. So the cell cycle is going to be interphase and then the mitotic phase. Now, mitotic phase is division. This is a cell, one cell becoming two cells. So the first part is called mitosis, and that's going to be all of these steps here. Prophase, this one breaks it into late prophase, but we're just going to deal with prophase, metaphase, anaphase, telophase. That is mitosis, which is going to be the division of the nucleus, so nuclear division. And then cytokinesis is actually separate because cytokinesis results in two different cells, so it's cell division. We have division of the nucleus and then division of the whole cell. And we're going to talk about each one of those steps later, but do know that where they fit into the cell cycle. Okay, we talked about most of this already. Okay, one thing to hit on here is that cytokinesis, it actually means division of the cytoplasm. Remember, the cytoplasm is everything inside of the cell. And so when we divide that, we're actually dividing a cell into two separate cells. This is a really cool picture that I like down here. It's going to result in two genetically identical daughter cells. So mitosis is just a copy machine. It's making an exact copy. It's like we took this cell and we made an exact copy of it on a copy machine, and now we have two cells. That's what mitosis does. So the division of the nucleus is mitosis, and the division of the whole cell is cytokinesis. Okay, so how does a cell know when to go through each step of the cycle? There are molecules that are called signals, a signal, and that's what's going to influence the activities of a cell. It's going to say, okay, move on to the next phase of interphase, or now we're ready for mitosis. And you don't need to know, I'm not going to ask you about all of these different types, just know that there are chemicals or molecules that act as signals. that will tell a cell that it's ready to go to the next step. Let me go back here and we can talk about those different stages of interphase. The first one is going to be growth, G1, and it's just a cell really just doing what a cell does. It's but it's also growing. So we're getting resources. We need resources in order to make another cell. And then we are going to do some more growth and also replicate the DNA. So we're going to have 2 copies of everything within our genome. We're going to have 2 copies of every chromosome, replication. We're going to talk about that in a lot more detail later in another chapter. And then G2, by the way, so we have G1, which stands for growth. This S here actually stands for synthesis because you're replicating DNA, you're synthesizing more DNA. That's S. And then you have G2, which is the second stage of growth. And final preparations for division. This is also where you're going to double the number of organelles. So each one of these phases of interphase, there are signals that will tell the cell, okay, you've grown a lot, now it's time to start replicating that DNA. So those signals are really, really important so that a cell knows when it's time to do the next step. There's also something here, see these red lines? These are called checkpoints. And these checkpoints are just so important because they're going to check and make sure that the cell is doing what it's supposed to be doing. So this first checkpoint here, it's going to check for cell size, nutrients, growth factors, DNA damage, because we don't want to replicate and make more of a cell that is broken. We don't want to make a cell that's not doing the job it's meant to be doing. So if a cell doesn't pass one of these checkpoints, then it should not divide. It should not make more cells. So this first checkpoint here is just seeing if it's growing correctly, if it's doing what it's supposed to be doing. If it's not, it can be kicked out into this thing called G0. And G0 is a resting state to see if maybe that cell can get its act together and grow and get more nutrients. And if it can, then it's able to go on to the next phase. Some cells actually just stay in G0 resting phase. It just keeps doing the job it's supposed to be doing and never divides. Example of that would be like nerve cells. Nerve cells, they don't divide. They just do their job that they're supposed to be doing. But once a cell, say a skin cell or a liver cell, once it gets enough nutrients and grows enough and is large enough, then maybe it can go on to the next phase, which is S phase, and do the replication of DNA and continue to grow. And then after G2 here, before mitosis occurs, there's something called the G2 checkpoint, and this one's going to check for cell size to make sure did it keep growing, and also did that DNA replication go the way it's supposed to go. So all this DNA was copied and replicated. Well, did that go as planned? If not, then we don't want to let this cell go into mitosis. And then there's also a check in mitosis here. And this one's going to make sure that mitosis is going the way it's supposed to be going. And we're going to talk more about that one later. So there are these checkpoints to make sure that what is supposed to be happening in the life cycle of the cell is occurring the way it's supposed to be. And if it's not, if something's going wrong with that cell, maybe it didn't grow like it was supposed to grow, or maybe it grew too much, it's way too big, or maybe the DNA wasn't copied correctly, then something called apoptosis might occur. Apoptosis is programmed cell death. So that sounds like a terrible thing, like, oh, we programmed this cell to die. But apoptosis is actually a very, very good thing because we don't want to make a whole bunch of copies of a cell that isn't doing what it's supposed to be doing. And I like the name apoptosis because it sounds like it's popping, and that's exactly what happens. The cell is going to pop and bubble into a whole bunch of different pieces, fragments. They're called blebs. It's going to break into little pieces called blebs, which is another really cool word. So it's going to pop. Apoptosis is programmed cell death. So it's going to fragment and blister, and then our white blood cells can engulf those cell fragments and dissolve them into pieces that we can use to make new cells again. Also, keep in mind, mitosis and apoptosis are the opposite of each other. So mitosis is going to increase cell numbers, where apoptosis is going to decrease the cell numbers. They're complete opposites, and they're both very, very important. Cell division and apoptosis are completely normal, and they happen in everyone. An abnormal cell that could become cancerous or a virus-infected cell are cells that should die through apoptosis. Now, when cancers or tumors get out of control, that's when apoptosis did not occur, and it should have. So sometimes apoptosis doesn't happen, or programmed cell death does not occur when it should have. We don't need to worry about this signaling protein P53. Just know that just like there were signals that would tell a cell to go to the next cycle, there's also signals that tell a cell to undergo apoptosis or to dissolve, and it stops the cell cycle when the cell's DNA is damaged. Okay, so this is just a review of the cell cycle here. We have interphase, a cell doing its job. We have these checkpoints, G1, G2, and the M checkpoint. Each one of these checkpoints checks to make sure that the cell is doing what it's supposed to be doing and can continue on the cell cycle. Or if not, then it needs to undergo apoptosis. There is something right here called G0. It's a resting phase where the cell can try to get its act together. Is it able to get enough supplies? Is it able to grow so that it can go to the next phase of the cell cycle? And then you have, let's say replication went well and the cell grew and did its job like it was supposed to, and it gets all the way over here to G2. So interphase went really well. It's going to check and make sure that that DNA replication went well, and then it's going to send it into mitosis, or the mitotic phase, which will include mitosis and cytokinesis. Remember, mitosis is going to be the division of the nucleus, where cytokinesis is the division of the entire cell. So division of nucleus, division of the cell. They're different, and they're both in the mitotic phase. Okay, next comes the structure of the chromosome, and we've talked a lot about this, so I'm going to kind of skip through it a little bit since I only have a few more minutes left on this video, and then I can touch base again next time. Don't forget, I think we've talked about histones. Histones are these proteins that the DNA can be wound around real tightly, kind of like your Christmas lights. When you take your Christmas lights off of your house, let's say, and you need to spoil them around something. You need to spool them around, like sometimes I don't have anything and I'm not very organized, and I might pick up a stick and start wrapping my Christmas lights around a stick. That's kind of the same thing the histones do. They wrap it up really, really, really tightly, and this only happens right before division. It's a way to organize the DNA. Okay, so normally, remember, our DNA looks like a bowl of spaghetti. It's called chromatin. Bowl of spaghetti. That's DNA that can be turned into proteins and do its job. It's unwound. It's found throughout interphase, just like this. But right before the nucleus divides, it has to organize all of this mess. So it's going to wrap all those like Christmas lights up into an organized fashion called a chromosome. So this is what the DNA looks like before nuclear division occurs, before mitosis. It's going to be tightly wound DNA around histones. It's found only during cell division, and the DNA in this form is too tightly packed to be used for synthesis, so it's just packed away that way. And I have to end the video right here, but I will pick it back up with the next video. Thank you guys. I miss you all.